Good afternoon, everyone. It's a real privilege to speak to you this afternoon. My research focuses on trying to develop new types of medicine for epilepsy. And I'm going to tell you today about one of the discoveries we made here in Dublin. I work at RCSI, the Royal College of Surgeons. And I'm going to tell you a bit about the journey from that discovery to where we are now, which is testing that medicine in dogs with drug-resistant epilepsy, natural epilepsy in dogs. And I'll tell you why we're doing that. There's a special reason why we didn't want to jump straight from the sort of the lab rat into humans. So I'll tell you a little bit about that story, and I'm going to talk a bit about genes and gene therapy along the way. So we've had a really good overview today of the different options for controlling seizures. Anti-seizure medicines, diet, electrical stimulation is becoming more of an option, and of course, brain surgery. We also know that 1 in 3 people will fail to respond to any of these options, and so we have this very significant need for medicines, new types of medicine. I'm not going to include too many graphs, but this is a really good graph, which I think most people can get, right? You can see that the lines on the graph are sort of all overlapping, right? What this is telling us is that if you are diagnosed today in 2024 with epilepsy, your chance of being seizure-free is the same as it was in 1984. We have many, many new medicines, but your chance of being seizure-free has not changed in decades. So we have to do better, and we have to think differently about the type of medicines we're going to use in the future. So I'm going to talk about gene therapy, and that sounds scary, right? But if any of you took a COVID vaccine, you had something a bit like gene therapy, right? And that'll become clear soon. You haven't got some permanent thing stuck in your body somewhere, changing your genetics. Don't worry about that. But as you'll see, the COVID vaccines were a little bit like a gene therapy. So the reason I'm saying that is just don't panic. This is going to be okay. Right. Before we get to gene therapy, what is a gene? Okay, maybe that's a dumb question. A lot of people in this audience will know a lot about genes and epilepsy, but not everyone. So let's start with the basics. Inside all of the cells in your body, including all your brain cells, is DNA. Right? We probably know DNA is the sort of instructions for life. Right? It's a very stable chemical, and it's a code. It's really information which, if read properly, tells the cell how to make proteins. See, proteins are really what cells used to do almost all the jobs. Proteins generate energy for cells. They generate neurotransmitters, which our brains use to communicate. They make the structures of brain cells. But a gene is really just a piece of that DNA that has the information to make a protein. But on its own, it just sits there. DNA just sits there inside the cell. For a gene to do something, we have to read that information. And this is what happens. Your DNA opens up. It's this little sort of what they call a double helix. It sort of splits apart, a bit like a zipper coming undone. And then an enzyme comes along, reads the information, and makes something called mRNA, which is a bit like a sort of a mirror image of DNA. That mRNA takes the information from the DNA into a different part of the cell, and in that different part of the cell, that information is now turned into essentially a protein. It's sort of read, understood, and we get amino acids, the little chemicals, glued together into a very long chain, and that becomes a protein. One way to think of this is, if you think of a recipe book, your Jamie Oliver recipe book, that's got the information, right, to make something, say, a cake. But essentially, on its own, it's just sitting there on a shelf. To make a gene do something, we need to bake our cake, okay? So the DNA is like the information, but the making the cake is the proteins at the end. Now, mutations. We've heard about genetic mutations. So in all of our cells, we have 2 copies of a gene. If you're told you have a mutation in a particular gene for epilepsy, that means there's an error somewhere in the gene. And what happens is that the protein that is made from that gene doesn't work properly. So as the information is read, and I'm going to show you an example here, you get the production of a faulty protein. Now, in this particular example, it doesn't really matter what the gene is. That faulty protein allows too many charged particles into brain cells. So brain cells are electrically excitable, right? And they send signals by a little bit like electricity. This particular gene mutation makes them too excitable. Too many of these charged particles come through, and now we have a more excitable brain. And this is one of the ways that a genetic mutation can cause an epilepsy. Now, everyone in the room is probably familiar with what a seizure looks like when they see it in their loved one or themselves. But I'm going to show you a little video which shows you what's happening in the brain when you have a seizure. So what I'm going to show you is a little patch of brain cells that have been stained with a chemical that glows when those electrical signals go very quickly. So the brighter they get, the more they go. So this is really what's happening inside your brain when a seizure happens. And it's going to start on the top right-hand corner. So you can see them almost pulsing away. They're communicating, brain cells do this all the time. And then boom, all of a sudden, right, there's massive increase in the firings of the signals. And it sort of spreads through the network, right? This is the seizure spreading within the brain. And then it dies back down again, it dampens down again. Seizures stop, right? The brain has some pretty clever ways to stop seizures. We just wish they knew how to do that faster, right? So gene therapy is simply administering genetic material, either DNA or RNA, to modify a gene product. So some of the COVID vaccines were a bit like this. We had mRNA in our COVID vaccine. That taught our immune system how to fight COVID. Now, what we're now seeing is that all sorts of gene therapies are emerging for lots of different human diseases. So this is where a gene is being altered somewhere in the body to cure or to treat or control a disease. This hasn't hit us yet in epilepsy, but there are now clinical trials of gene therapies for some of the epilepsies. This is an example of one of them. This is a gene therapy for Dravet syndrome. Dravet syndrome is almost always caused by a mutation in the SCN1A gene. So this particular gene therapy in trial basically boosts the amount of that gene being produced within brain cells. This is going to be game-changing for people in the future if this works out, if this is safe enough. We don't know that yet, but we think it will be. Here's the problem. Many epilepsies, more than half, are probably not caused by one gene. Maybe some effect of small effect of lots of different genes, or it may have nothing to do with your genetics at all. It may be because of a head injury, right? We can get injuries to the brain. Anything that damages the brain can cause epilepsy to develop. This is a brain scan of somebody with temporal lobe epilepsy. We've heard about this earlier. Sometimes there's damage to a part of the brain, and if it's drug resistant, you may go to a neurosurgeon. This is Donick O'Brien at Beaumont, where he may remove that bit of the brain that keeps causing the seizure. This is a piece of your brain if you've had brain surgery. Now, the amazing thing about brain surgery is it allows scientists to look into the brain tissue and to learn a bit about what is different in the part of the brain causing the seizure. This is a piece of brain tissue, the little brown dots, those are neurons in the brain. Here's the problem. Lots of things are different within this part of the brain that's causing the seizure. I won't go through them because it's not important. The take-home message is this. There's not just one gene to fix. So, with many, many of the epilepsies, particularly epilepsies acquired as a result of a brain insult, there isn't a way to just go in and fix one gene. We need another approach. We need a way to actually fix or adjust, is maybe the right word, lots and lots of different genes. And if we can rebalance those genes towards what they should be doing normally, we may be able to fix more of the epilepsies. So what is causing seizures in that type of epilepsy? Well, it's generally a change to the structure or the function of brain cells. Well, what caused that? Changes and differences in gene activity. So that's when the gene switches on or off. Not the DNA, we're not talking about changes in DNA. What we're saying is some genes switch on and some genes switch off in the brain that causes seizures. So really, what one of the drugs of the future will be is something that can adjust gene activity. Not the DNA, but the activity of the gene, the baking the recipe part, right? So here's an illustration of how gene activity changes with seizures. This is an experiment I ran a very, very long time ago in the University of Pittsburgh. We exposed a rat to a seizure. There's certain chemicals that can cause a rat to have a seizure, and then we looked at genes that switched on in the brain, and you can see between the control, where it's this light grey colour, and then after a seizure, this dark stain. This is basically active gene activity in the brain that's been stained with radioactivity. But you can also see that within a few hours, that gene activity goes back to normal. By 3 days, 72 hours, that gene is switched off again. So genes switch on and off all the time, and this is what we are trying to do in our lab to try and control that on-off process. But, coming back to that point, the problem is there's hundreds of genes where we see small differences in their activity. How do we control that? Our new therapy would need to control hundreds of genes. How can you do that? Well, you need some sort of a master controller, and such a thing exists. Now, anyone know, the picture on the left is the Earth a long time ago, and the picture on the right is what life looked like on Earth at that time. Anyone want to guess how long ago this was? 20 million? Any more? 20 million?
Who took the picture?
It might be an artist's impression. All right, in the interest of time, that's 600 million years ago. So that's the Earth 600 million years ago. Very, very early on in life on Earth, a new type of gene emerged. It didn't code for the proteins. What that new gene did was produce short RNAs. That's the little chemical that sits in between DNA, our code, and protein, the doing thing. Short RNAs. And those RNAs ended up with the name microRNA. MicroRNA do a very important job in every cell in your body right now. What they do is they dampen down gene activity. They are nature's stabilizing system. So gene activity is actually quite tricky to control for cells. It kind of happens in bursts and starts. And this other type of gene, this microRNA, evolved a very long time ago to stabilize how our genes are switched on and off. Now, I was trying to explain this to my dad. My dad's an engineer. And he said, the Tacoma Bridge. I was trying to think of some way to get this across, right? So nobody's going to know what I'm talking about. But in the 1940s, an incredible new bridge was built in Tacoma, Washington, the USA. And it was a complete engineering disaster. So this is taught at engineering schools. But it's a great analogy for thinking about why we need to stabilize gene activity, okay? This is the... Oh, sorry. The dramatic.
Dawn of a fatal day, and the wind begins to speak with a roar that no man can fail to hear.
Bridge. This is a huge bridge, and it's just twisting and turning up and down.
In a 40-mile-an-hour gale, the feather span weaves like a ribbon.
And this is what your genome would be like if you didn't have microRNA. It would be going too high, and then too low, and then too high, and then too low.
There's an automobile caught on the heaving roadway.
So this is what life would be like without microRNAs, as an engineer would see it. Okay, and I'll skip ahead in the interest of time, but the bridge collapses at the end, so check it out on YouTube, it's a bit of fun. Now, the kind of super cool thing about microRNAs is that they also do this by sticking onto lots and lots of other genes. They kind of regulate a whole group of genes at once. So now we've sort of got our solution, right? We've got our big problem. Lots and lots of genes are differently active. How can we do that? You go after a microRNA. You find the master controller, and then you can adjust gene activity. But you have to find the master controller, right? So this is what we've been doing in the lab in Dublin for a long time. We start with mice. I can talk about why we do that later. We take a little bit of the brain tissue out of a mouse that's had seizures or epilepsy, and then we can look for these microRNA genes. And we spotted one that always went up. It got more abundant. There was more of it in the brain in mice with epilepsy. And then we checked. We chatted to our neurosurgeons at Beaumont. They had some brain tissue donated by patients who'd had an operation. And the same microRNA exists in humans, and it also did the same thing in epilepsy. So maybe this is good, right? But how do you block a microRNA? Well, you need a completely different type of drug. And earlier, Sophia mentioned antisense oligonucleotides, right? If you didn't know what that was, that's basically an artificial form of DNA. So basically, we can make little versions of DNA in the lab, and then those we can design to stick onto our gene. So it's very, very precise, very, very precision type of therapy. So we can design this to our microRNA, when we can now stop it from working. So what happens if you stop this microRNA working? We've tested this in mice with epilepsy. So in this model, mice begin to develop epilepsy because of something we do, which I can talk about later if there's time. And then we inject them with our microRNA inhibitor, our blocker. So what you're seeing in this graph, there's a placebo group, that's the red line, and the blue line is the treatment group. And on the y-axis, that's how many seizures per day the mice were having. So they start having seizures, and then after 2 weeks, 14 days, we inject them with the microRNA therapy. And you can see within a couple of days that the seizures start to drop, and they drop really, really low. And we've studied mice for a couple of months afterwards, and they have about 90% less seizures. So this is really exciting, right? Let's go straight to people. Or maybe not. Maybe we need to take a pause, right? Because this is a very different type of medicine. This is not your small pill that you can take 2 or 3 times a day. This antisense, once it's in the body, takes a long time to break down, 1 or 2 months. So if you target a gene with antisense and something goes wrong, it's stuck there for 2 or 3 months. So we have to be very, very careful. So it was this thinking that made us ask 2 more questions. First of all, are the genes that are controlled by the microRNA, are they the same in humans or larger animals at least? And are they safe and effective? Would they stop a seizure in a larger animal? And this takes us to dogs with epilepsy. So we were thinking, what could we do between the mice, but before we go to humans? And it's been known for some time that dogs naturally have epilepsy at about the same frequency as humans. We also treat dogs with epilepsy with the same drugs that we treat humans. So the frequency is about 1 in 100 dogs. And actually, some breeds, the frequency is even higher. And about 1 in 3 dogs with epilepsy stop responding to treatment. They're drug-resistant dogs. 54,000 is our estimate in the UK. I'm not sure what the numbers would be like in Ireland, but equivalent. And we give them drugs every day, sometimes 2 or 3 times a day, to stop them having seizures. So they don't control seizures all that well, similar situation to humans, and they have a lot of the same side effects. And, as vets will tell you, vets would also love to have new treatments for dogs with epilepsy. And so if you look at, if you think of it, this as a way to test therapies, particularly something like a gene therapy before you go to humans, okay, you've got epilepsy that can be caused by injury to the brain or by genes. Dogs with epilepsy have similar looking seizures. Sometimes they're focal onset, sometimes they're generalized straight away. And they have some of the same comorbidities. And then, of course, we use some of the same types of drugs. So dogs with epilepsy could be a way to test things like gene therapy, which are a bit scary to give to people straight from a mouse. So we'd been working on this microRNA drug for a while, and then I got an email from a veterinary neurologist. So this is a neurologist that specializes in looking after dogs with epilepsy, called Rodrigo. He's at the University of Glasgow, and he said, I have some terribly sick dogs, and often these dogs have to be put down. And would you like to test your microRNA drug in our dogs with epilepsy? So I'm going to show you a video of a dog having a seizure. This is one of the dogs that was enrolled in the clinical trial. It will be upsetting to some of you. So if you feel that you would be upset, do not watch the video. I can't stop you from hearing it, though. So this is filmed on an iPhone. So the dog has just started having a partial or focal seizure, and it's going to generalize in a few seconds.
Good boy. All right, okay.
Very good.
Okay.
Okay.
All right, okay.
Baby. Okay, baby.
It's okay. It's okay. It's okay, baby. Let's get back. It's okay.
It's horrible stuff, right? And if anyone has a dog, that has epilepsy, and there's a possibility, it's absolutely destroying for them, and oftentimes the animal has to be euthanized.
I have one, but it isn't that violent.
Okay. So, after having talked to the vet, we said, well, the first thing we need to know is the microRNA, is this same gene really present in dogs, and is it altered? So he sent us some samples from dogs with epilepsy, some that had quite controlled epilepsy and some that didn't. And the level of the gene was increased in the dogs with the drug-resistant epilepsy. This was a good start. And then he had some brain tissue samples from dogs that had been put down, and that the owners had donated their bodies to charity, to science. And when you look in the brain, the little black dots, the little black circles, these are brain cells making the microRNA. And so generally speaking, when we looked at the results, it looked like the dogs with epilepsy looked a lot like the mice and like the humans. So this led us to METRIC, which is our clinical trial of our microRNA therapy in dogs with naturally occurring epilepsy. And there's two simple aims. First of all, is it's safe and effective and non-toxic. These are our inclusion criteria. So these are adult dogs that have failed at least 2 anti-seizure medicines that were all having about 4 seizures or more per month. Some of the dogs have far more seizures than that. And this was run as a double-blind, placebo-controlled, randomized trial, so that the vet doesn't know what he's giving. He's just handed a tube, and the owners, importantly, don't know what their dog has been given. And this is just an overview of the trial. So all of the dogs who are entered, we record EEG, we take blood samples. It's run very much like you would run a human clinical trial. And then we follow the dogs over time at different times. And if the seizures change, particularly if they get worse, then the doctor is allowed, the vet is allowed to what's called unblind, find out what the dog was given. Because, of course, our biggest fear was we would give this microRNA drug to the dog and something terrible would happen. We didn't know. So we're looking at safety, and then we're looking at efficacy. Ideally, what we'd like to see is that seizures would drop by more than 50%, because taking any kind of gene therapy through to human trials, you'd really need to know that it would do something important, significantly change and have an impact. So I'm going to show you the results of the first 11 dogs. So we have 5 dogs that were given the microRNA therapy and 6 that were on placebo. The 2 groups are well matched in terms of different breeds of dogs, their age. There's a mixture between male and female. They've had epilepsy for about the same amount of time, and they've all been trialed with multiple anti-seizure medicines and have failed. They're all continuing to have seizures. This is possibly the most important data of all, that it appears to be safe. So the main side effect that you see when you inject this, and it's an antisense oligonucleotide, this type of gene therapy, the main side effect you see is a slight increase in body temperature. They get a bit of a fever, and they have some neck pain. And that's from where it's injected. But the vet gives them paracetamol, and they're fine the next day. So this is really good news for us. And then these are the most important findings of all. Mainly, if you look at the right-hand side, this is the graph. So what this is showing is the seizure reduction. So 0 means it has no effect at all. And then the blue bar, that's the treated group with the microRNA drug, and it's below the -50. So 4 of the 5 dogs had a better than 50% reduction in their seizures. And as you can see with the placebo group, most of those dogs continuing having seizures. Some of them even had more seizures. In fact, most of the dogs in the placebo group were unblinded. That meant that the vet was concerned that they were having more and more seizures, but it was actually the placebo group. And I'll show you data from one individual animal, which is really striking. This is our super responder dog. So on the left-hand side, before the red dotted line was the baseline. So this is how many seizures the owners counted the month before starting the trial. And it was nearly 200 seizures in their dog. The month after they'd had the injection, they had no seizures. The month after that, they had no seizures. By 4 months, though, the seizures came back towards how they used to be. And so the vet gave a second injection, and this time, they didn't have a seizure for 11 months. This is the most up-to-date data we've got now. So this is really, really exciting for us. Not all the dogs have responded as well as this, but it does suggest that you can have very long-lasting control of seizures with this gene therapy. And this is the little testament. This is the picture from the start. The difference from, and the super responder's dog is Oscar, was before treatment, is now amazing. It's been incredible to see Oscar, who is otherwise healthy, be back to being a normal and happy dog. It's been a revelation. If we hadn't got Oscar on the trial, he 100% wouldn't be here. So, in summary, our approach, our targeting of this gene called microRNA represents what we call a network therapy, something that can change all sorts of genes within the network in the brain that causes the seizures. The injection of the microRNA therapy is well tolerated with mild side effects, and it has long-lasting anti-seizure effects. We're continuing to recruit cases for this, trying to understand a little bit more about how the microRNA drug works, and we've begun testing this in human models. So there's a couple of ways that we can test this in a sort of a human model that isn't an actual human, and I'm obviously happy to describe that in a bit more detail. Many, many people have worked on this over the years. Some of the names are here. Christina Reshka, who spoke earlier, was in my lab for a while, and the vets in Glasgow are just incredible people. And if you have any interest in this topic, I've just published a book called Fine-Tuning Life. So you can read that, and it actually does mention a little bit about the dog trial, although when I wrote the book, the dog trial hadn't started, so I didn't know that it was going to work.